Hello everyone. So what we're going to discuss in this lecture is how do living organisms protect themselves against foreign invaders, so organisms that do not belong to their body. So we'll talk about different things. This is just the outline. So first we'll talk about the different defensive mechanisms, which are basically three of them. The first one is there are natural barriers that do prevent the entrance of different organisms inside the body. So these are referred to as epithelial membranes. We'll go quickly through that. Then we'll talk about what we refer to as the actual immune system, which consists of two parts, the innate immunity and the adaptive immunity. So we'll talk about the fighting strategies and so on. In particular, we'll spend more time talking about the adaptive immunity, the types that you have, the antibody-mediated and cell-mediated, and how this leads to selection, specificity, first of all, selection of cells and the formation ultimately of memory in the immune system. And ultimately, we will finish it up also talking about how do we end up having all that variability in the adaptive immunity so that you can actually recognize so many different types, specifically so many different types of pathogens. So starting about the immune system here. So in general here, there are different defense mechanisms, as we said before. We have three main types. First of all, there's an epithelial barrier, which we all have. And this consists not only on your skin, but also on your guts, your respiratory lining, and so on, which is already preventing most of the organism from entering your body. And once the organism enter your body, you have two different types of response, which is the innate and adaptive immunity, which do ultimately prevent the organism that have entered from spreading in the rest of the body, or most of the time. Sometimes it doesn't work. Not all organisms have both the innate and the adaptive immunity. We'll talk about that later on. So talking about the first defense, the epithelial barriers. Now, there are many barriers there. Obviously, your skin, it's one example, where you have your cells that are nicely, tightly put together through tight junctions so that bacteria or viruses cannot go through them. So there are multiple layers, and also the way they're bound together, the cells, do prevent the entrance of the organism. Together with that, you have other membranes as well, like the one found in your lungs, in your guts. So mucous membrane that do prevent the entrance of the organism, not only by having the cells themselves tightly bound, but also by producing mucus. If you remember when we talked about the bronchi, we were talking about how they produce mucus, and together with the cilia that they have, they actually move this mucus outward. That's an example of defense mechanism. And also the acidity of the stomach, the digestive enzymes, different enzymes that produce, like lysozymes, for example, your saliva contains lots of them, and the presence also of antibodies thrown in different fluids already prevents the entrance through the epithelial barriers into your body or any organism body. The second type of immunity that we have, or the defense, is what we refer to as the innate immunity. Now, this is present both in invertebrates and vertebrates and plants, and differently from the acquired immunity, which is the one we normally talk about, the one which is cell-mediated and antibody-mediated. So the innate immunity, differently from the acquired immunity, is non-specific. So it fights anything that comes in. Its activation is non-specific, is not caused by the presence of a specific pathogen, but it just reacts against any pathogens. And as a consequence, it does not produce any memory. So it's not that if you get exposed to the same bacteria twice, the innate immunity will get any stronger on the second type. It's always at the same level. Now, inflammations, we'll talk particularly about inflammation, is one of the innate immunity mechanism that is very commonly used both by invertebrates and vertebrates. The third type is what we refer to as adaptive or acquired immunity, which can be both cell-mediated or antibody-mediated. Differently from the innate immunity, in this case, this level of defense is triggered by specific microorganism, and it leads to memory. So what it means, and that's the base of the production of vaccines, is exactly that. If you have an organism that it infects you once, if that's the first time, you would have your adaptive immunity acting on this organism, not only removing it, but also acquiring memory so that the second time you get exposed to the same organism, you will get, again, the adaptive immunity to react, but in this case, in a much stronger scale, so that the organism can be removed much faster before any damage can be caused. Okay, so when we talk about immune response, in general, what we're talking about is both the innate immunity, particularly inflammation, and the adaptive immunity. So the two different types, second types that we talked about, and that which are actually connected with each other. You see when we talk about inflammation, how the cells that do actually are part of inflammation, so the innate immunity, are also the same cells that do help the stimulation of the T and B cells in the adaptive immunity. Now, no matter what type of mechanism you're talking about, it always involves some sort of recognition with pathogens. And this is done by some cells. You see macrophages or dendrocytes are cells that then are referred to as antigen presenting cells, or APC, as you can see here. These are cells that do recognize foreign organism compared to organisms that do belong to the body, or cells that do belong to the body, by recognizing a pattern of different molecules that are unique of cells that do not belong to the organism. These are referred to as pathogen-associated molecular patterns, or PAMPs. Basically, these are different molecules, for example, glycoproteins, that are found uniquely on cells that do not belong to the organism. For example, there are some glycoproteins in the membrane or the cell wall of bacteria that are not present in your body. So if some of these cells sense the presence of several of them, a pattern of them, now they know that they have to act against that. Okay, and they do that by the use of receptors. You can see here this blue structure here will be receptors that do ultimately sense the presence of this molecule. When they sense that these are molecules that do not belong to the body, again, it's a combination of them, then they get activated by causing the innate and eventually the adaptive type of response. Now, in terms of non-specific defense, so we have the innate immunity uses different strategies there. First level of strategies, you see when we talk about inflammation as well, is phagocytosis. So there are some cells, macrophages, dendrocytes, that do actually engulf the bacteria or the viruses that do eventually enter the body. Another way this is done by insect is melanotic encapsulation. It's basically the secretion of proteins that do form a capsule against the foreign organism. And a third way to react without any specificity is by producing peptides that do act against the microbes that I mentioned. And so an example are defensins. These are proteins that basically produce and they have antimicrobial activity. They are produced by different cells, including macrophages. And what they do, they basically enter the membrane of the attacking organism. And when they enter there, they start forming pores. So that now you have that the cytoplasm of these bacterial, or whatever they are, so bacteria in this case, it enters the bacteria, causing the cells to die. Now let's go through inflammation. So inflammation is a reaction which is part of the innate immune system. So it's a non-specific type of reactions. It's characterized by several properties, which is the presence of swelling, usually, redness, heat, and pain. All of them basically caused by the different molecules that are released during inflammation, and the way it works by basically trying to attract as many cells by causing the capillaries to become more leaky. So how does it work? Well, the first step in inflammation is the detection by cells called macrophages. These are cells that are found in your interstitial fluids. They are formed by monocytes. Monocytes are cells that are always in circulation in your blood. So they're blood cells always in circulation there. They tend to stay there. After 2-3 days, usually they tend to home in the interstitial fluid. No matter whether there is inflammation or not, they end up there. and they turn into macrophages. And so these are the cells, the first cells that do sense if there is anything. You remember the PAMPs, the pathogen associated pattern, is that sense if there is a pathogen that is in the interstitial fluid. If they detect a pathogen, what they do, first thing they try to do is to actually phagocyte the pathogen. They try to ingest it. Many times this is enough. If you have very little pathogen, this is enough. Okay, inflammation basically ends there. But now at the same time though, usually what they do though, so they release other molecules, cytokines and chemokines, which have the function to basically escalate the reaction in case it's needed. So cytokines, what they do, they cause the swelling, and at the same time, they also end up in the bloodstream where they actually recruit more immune cells on the spot where the pathogen is sensed. Chemokines do a similar thing, so they actually recruit these immune cells, but what they do basically, they direct them to the spots where they should go. So how do they work is, let me just turn on the pan. I'm trying to turn on the pan here, if I can do it. There we go. So imagine this is your skin. This is where the pathogen is. And this is your capillary here. So now you have macrophages that do try to ingest it. They release chemokines and cytokines. The chemokines would have a concentration gradient, which gets lower and lower and lower toward the blood. So if you have any cells that here sense the cytokines, neutrophils will do that. They end up exiting the capillary here, and then they will follow the concentration gradient, knowing exactly where to go, where the bacteria is present. So macrophages are the first cells involved in that. Now you see that these cells, later on, they're also those cells, together with dendrocytes, that do become antigen-presenting cells. Keep this in mind. This is part, now will be part of the adaptive immune system. So now you have that the macrophages have been activated, they release cytokines and chemokines, and then there are other cells that are now attracted there, which are mast cells. Mast cells and basophils are similar cells. They're different cells, but similar. They work similar way, sorry. And what they do, they sense if there are dead cells. It's possible that the pathogen is actually killing also the macrophages. And so these cells get activated by that. And what they do, they release, first of all, histamine. Histamine is what ultimately, together with cytokines, causes the dilation of the blood vessels, of the capillaries. So basically, this is getting the capillaries ready to let more cells move into the affected area. Mast cells, they release also heparin. You don't want having the formation of any clot in that area. You want the cells to be able to flow in there. So heparin prevents clotting. And so together with that, now you have the cytokine produced before. And what they do, as we say before, they actually do attract more immune cells, particularly neutrophils. They are circulating in your blood. And they're going, basically, by doing that, they attract them. So they sense neutrophils, they sense the presence of cytokines, and they know where to stop in the blood vessels and exit the capillaries and get into the interstitial fluid where inflammation is taking place. And then following the chemokine gradient, as we say before, they will be actually going exactly to where the bacteria or the pathogens are found. Now, macrophages and neutrophils, they try to phagocyte these pathogens also, but also they release also lysozymes, so enzymes that do act against bacteria, and defenses too. Do you remember those proteins that form the pores in the cell membrane? Many cells die, and that's so neutrophils particularly, and that's exactly what caused the formation of pus. Hopefully you're not having lunch while you listen to this. Together with that, now you have more cells being attracted there. They're producing more and more enzyme. Now the swelling, the redness, the heat, as we say before, which are typical of inflammations, are caused by the action of both histamines and cytokines present there. Now, together with that, so all of these leads to the big response, cell-mediated response, ultimately against the pathogen. So now you have macrophages, dendritic cells, you have neutrophils. They're all trying to get rid of these bacteria in different ways, by phagocytosis, but also by trying to produce lysozymes and so on. Sometimes this doesn't work, and this basically leads to the movement from inflammation into the third type of mechanism, defensive mechanism, if you remember, which is the adaptive immunity. And this is done by macrophages, and dendritic cells, which do ingest these pathogens. And when they do ingest them, they basically break them down into molecules referred to as antigens, which then they will present to the cells of the adaptive immune system, so B cells, T cells, and so on. And by doing that, they work as antigen presenting cells, which then will activate these cells at different lymph nodes. Now, in terms of cells that we talked about here, so all the cells we mentioned before, they're all blood cells, okay? So they all are formed by the hematopoietic stem cells, which, if you recall, when we're talking about different tissues, are located in some of your bones. So these hematopoietic stem cells give origin to all cell types. First of all, they produce two types of stem cells, the myeloid stem cells and the lymphoid stem cells. Now, the cells that we talked about until now, they're all myeloid cells. So we talked about mast cells, which are very similar to basophils, the way they work. So they're all rich in histamines. These granules you see there, they're all containing histamines, which then get released. Then we talk about neutrophils. We talked about macrophage and dendritic cells. So these are what will turn into antigen presenting cells, which then you see later on are those that do activate T cells and B cells later on. Okay, so these are the cells that we're talking about here. So this is just a schematic that summarizes what we just said. So you have that somehow a foreign organism has entered your body. Okay, so your first level of defense, the epithelial barrier, has failed. This could be a splint, for example, right? If you're working with wood, bring some bacteria in. And the first thing you get is macrophages, trying to get rid of them by ingesting them. At the same time, they produce cytokines and chemokines, right? Which do recruit neutrophils from the blood. This is helped by mast cells, basophils as well, that do release histamine and other molecules that do cause these blood vessels to become more leaky and so on. Cytokines as well do that. You have neutrophils. Now they go there, they try to ingest the bacteria as well, dendritic cells, and then some of these cells, macrophages, dendritic cells, and so on, they'll actually go back to the circulation. And they'll end up, for example, in your lymph nodes and other parts of the lymphatic system, where they will be activating now the next level of defense, which is the adaptive response. Okay. These are just a summary of what you say. Together with that, we have other systems that do act in the innate, as part of the innate system, and some of them are what we call the complement system. This consists of about 30 different proteins that you have normally circulating in your blood. They're usually inactive, but then they get activated by different antibodies that are going to be sent there as well. And these basically, they do act by coating the pathogens, okay, and killing them. Some of them also form the formation of pores, and therefore they cause the lysis of these different organisms. Again, they're non-specific in this case, so a part of the innate system. Now, the response when it comes specifically to viruses can be slightly different. There are other mechanisms, innate mechanisms, that are involved in there. So cells, they express short RNA molecules called short RNA, shRNAs, which do act by RNA interference. And basically what they do, they can destroy the RNA of some of the viruses. Other ways that they work is by producing cytokines. So the infected cells, sometimes they produce cytokines. So imagine this cell being infected by a virus. They can produce cytokines called interferons that do act on their own or nearby cells too. They could be infected as well, So they can act autocrinally or paracrinally, as we said when we were talking about the endocrine system, if you remember. And by doing that, they inhibit basically their own protein system, synthesis, and therefore the one of the virus that infected them as well. Other ways, some cells, they cause themselves to die. They just activate a mechanism of cell death referred to as apoptosis. They sense the presence of the virus and they basically do a kamikaze action. They kill themselves. By doing that, they kill the viruses too before they replicate. And then also there are other cells, natural kill cells, which are lymphocytes, that what they do, they sense that and they produce molecules that do actually kill the infected cells by basically making holes in the cell membranes. Now let's move on to the last type of system that do prevent the invasion of foreign organism, which is the adaptive immunity. So we talked about the epithelial barrier, the innate immunity, and now we talk about the adaptive immunity. Now, this is a specific, if you remember, differently from the innate immunity, it's a specific type of response. So it's triggered by a specific presence of certain molecules, which we call antigens, and then they lead to the formation of memory. So the event will be remembered for the future so that a response could happen again in case the same pathogen enters with a higher degree of response. And this is mediated by two types of lymphocytes. So these are lymphoid cells. If you remember, the hematopoietic stem cells can produce two stem cells, the myeloid stem cells, which will be the one that produce the macrophages, the dendritic cells, and so on. And then you have, they can produce lymphoid stem cells. This is the stem cells that produce all of these lymphocytes we're talking about right now. So you have B cells and T cells. Their naming comes from where they mature. The B cells do mature in the bone marrow, whereas the T cells, they mature in the thymus. And then from there, they move to different parts of the lymphatic system, so particularly to the lymph nodes. That's where they will be detecting the antigen, and that's where they get activated to respond. This summarizes what I say. So what you have here is that the stem cells that are present in the bone marrow, they do produce this immature lymphocyte by producing first the lymphoid stem cell. Then you have that the immature lymphocyte in the bone marrow, they mature into B cells, whereas in the thymus, some of them move through the blood to the thymus, and that's where they mature into T cells. They both move to different parts of the lymphatic tissue, particularly lymph nodes, and that's where they will sense and they will eventually get activated by specific antigens. Now, there are two types of adaptive immunity. There is an antibody-mediated immunity, which is the one that is basically done by producing proteins called antibodies. And there is one which is cell-mediated immunity, where instead the action against the pathogen is done by cells, specific cells activated by the specific pathogen or the antigen in the specific pathogen, and then these cells are the ones that directly kill the infected cells or pathogens by acting directly, not through proteins. Okay. Now, no matter what type you're talking about, the steps are similar. You have the first step is that there is an encounter of the lymphocyte with the antigen carried by the pathogen. This is mediated by antigen presenting cell or APCs. And then you have that the lymphocyte get activated. Whether it's a B cells or a T cells, they get activated. Some ways this leads to the removal of the antigen and the pathogen, which has entered. And at the same time, it leads also to the formation of memory through the formation of memory cells. So the steps are the encounter with the antigen, the activation of the lymphocytes, the removal, and finally, at the same time, the formation of memory against that specific antigen. Now, the encounter with the antigen, which is presented by macrophages and dendritic cells, the antigen presenting cells, those cells that were involved in inflammation as well, if you remember, that do phagocyte the bacteria or the viruses that enter. So these encounter, so basically the antigen presenting cells, they do break down the foreign organism into small molecules, and then they do present them to the lymphocytes, B cells or T cells, through specific receptors, which are called B cell receptors if are found in B cells, or T cell receptors, which are if we're talking about T cells. Okay. Now, you have to imagine one thing. There is a huge variability of these receptors. We'll talk at the end on how this variability is generated, but they all act in a very similar way. But you have this huge variability, meaning that you have this huge number of B cells or T cells that can recognize all different molecules, okay? So all different antigens. So just to give you an idea, you have about 1 trillion, B cells there, okay, that can recognize basically so many different type of antigens there. So you have cells basically that can recognize almost any possible molecule. So, and therefore any possible organism that can enter your body. How is this variability generated? We'll talk about that at the end. Focusing on the receptors here, B cell or T cell receptor, that's the way they look. So on the left, you have a B cell receptor, which is formed by two type of chains duplicated. So you have two heavy chains, okay, and two light chains. Now, when it comes to the heavy chains, this has a transmembrane domain. That's what anchors this receptor to the membrane. It has actually a domain this side as well. And then it has a constant region here. This is the same in all B cells, okay? And then it has a variable region here at the end. When it comes to the light chain, you have two of them identical to each other. They both have also a constant region, and they have a variable region here at the end. Now, is the variable region here, which is different from B cell to B cells. And this is the one that makes this receptor specific for a certain molecule. So for these will recognize, let's say, a specific part of a molecule coming from a coronavirus, for example. But in another B cells, this will be specific for another molecule from coronavirus, or will be specific for another molecule from a different type of viruses. Okay, so that's the region here, the variable region, which is different. Again, you have trillion of possibility there, which makes the B cells different. When it comes to the T cell receptor here, the structure is simpler. It's formed only by two peptides here, which do have still a transmembrane domain here, a constant region, and now they have a variable region at the end again, which makes the T cell receptor specific for a specific antigen. Okay. Now, the antigen is any molecule belonging, any molecule if you want, that can be sensed by these receptors. Now, when it comes to these receptors here, these variable regions here, so the receptor there will sense only specific part, will be specific for a specific part of an antigen. So imagine this antigen being a glycoprotein in a pneumonia bacteria. These, you would have that some receptor do recognize this part right here. Okay, so this is within a B cell. Some others do recognize these other parts. We're talking about 10 to 15 amino acid part of a protein, just to give you a sense. So the same antigen can actually be recognized by different B cells through different B cell receptors. Okay, the same thing for the T cells. So they're so specific for different parts of the antigens. Now there are different types of antibodies focusing on the antibodies produced by B cells here. There are different types: IgM, IgG, IgA, IgE, IgD. These are all immunoglobulins, where the Ig stands for. If you remember, they are one of the components in the plasma, the protein component of the plasma of the blood. So you have different types. You have IgM. These are the first type, and the difference between them is ultimately in the constant region, which makes them either polymerize like the IgM or not, or act on their own. So this is basically what will be the receptor, the IgD. Okay, so this is the surface receptor that you have in there. Now this, once the cells gets activated, it can produce first thing will produce IgM and IgG. So in the first response, that's what you produce. This is produced faster, okay, the IgM, and this usually stimulates the reactions of some of the cells, even in the innate immunity, to act against the pathogen. The IgGs that are the most abundant one, antibody produced after the stimulation of a B cells. Okay, so these are the real big, if you want, actors in the antibody-mediated response. And then you have some other ones like IgE. They are normally produced. They are non-specific, so they're just produced all the time. For example, by your bronchi, by your GI cells, your skin, and so on. The IgA2, they are constantly produced. They're non-specific, constantly produced in the milk, in your saliva, and they basically give a basic protection on the outside of your body against most organisms. Okay, that's one of the reasons why, for example, if you cut yourself and you don't have access to anything to clean yourself, a good idea is actually sometimes to lick that cut. Is the reason being that if you lick it, you're actually putting some IgA plus other enzymes, lysozymes, that could eventually kill organisms that are present there. That's what animals do. Dogs, they lick their cut, right? Their cuts that they have around their body. So moving on here, now you have these receptors, so B cell receptor or T cell receptors, that are the ones that do find, that basically meet with the antigen. And now you have that these will lead to the activation of the two type of cells, T cells or B cells, which are part of the adaptive immune response. So looking at the antibody-mediated immunity here, what is the next step? So what happens there is that you have some cells, the dendritic cells or the macrophages, that do actually migrate during inflammation. So they engulf the pathogens, they break it down, and they do migrate through the blast system. They do migrate to a lymph node, okay, or the thymus there. And there, basically what they do present pieces of the bacteria or the viruses broken down. This will be referred to as antigens, together with a certain type of molecules called the class 2 major histocompatibility complex. They do present it to the T cells. These are a specific subset type of lymph nodes cells. So that's how they do it. They have engulfed the bacteria, they break them down, and now these antigens here are presented to the class 2 major histocompatibility complexes here. They are presented now to the T cells that are present there. Now, the type of T cells that they do activate are referred to as CD4 T cells. The reason being they're called CD4 because they present this protein called CD4. Excuse me. Now these ultimately, so these CD4 cells, they do bind to the antigen with the T cell receptors, and by doing that they get activated. This binding leads to the antigen present cell to produce interleukin, so the cytokines, which then stimulates the T cells that are bound to them through the T cell receptor to be activated and expand. So the cell that's bound now to these antigen presenting cells will start replicating, getting more and more and more. So by doing that, basically you end up having thousands, if not millions, of T cells which are specific, that have the receptor specific for that antigen, that start being produced. Okay, these cells are referred to as helper cells, and these are cells that do not act directly, but what they do, they will stimulate the B cells. So this is summarizing what I just said. You have the antigen presenting cell, so this will be a dendritic cell, has engulfed the bacteria, for example, here, or the virus, and now is presenting parts of this bacteria to the major histocompatibility complex 2 to the T cells. Okay, this leads to the formation of interleukins, which then stimulates these cells, the CD4 plus the CD4 receptor is necessary here to expand and become helper cells. As the name says, helper cells are cells that do help now the B cell activating the B cell. So how does it work here? So now you have that the B cell, the complex here. So now you have that D, going straight to this picture here. You have that D T cells. Now they have the specific receptor for the antigen here, right? So they do actually bind now to this antigen here, and now they do present it to the B cells. Now these will be B cells that do have, again, the B cell receptor specific for that antigen. So ultimately, these are cells that have a receptor that's specific for that specific bacteria or virus origin, right? This leads again to the production of interleukins, in this case by the T cells, which then stimulate the B cells to expand. So now you have millions of B cells with the receptor specific for this antigen being produced, right? These cells, they lead to two things. They lead to the formation of plasma cells. These are the cells that start producing the antibodies, which then will fight against that antigen. And some of these cells, they just stay quiescent and they're kept as memory cells. So if these antigens comes back, let's say months later or years later sometimes, now you have more of these cells present in your immune system ready to fight it. How do you see that? You see it here, for example, if you measure the amount of antibody produced in the blood through a first exposure and a second exposure. So imagine here on the left, you see how it will be the response the first time you're exposed to a certain antigen. So this is the amount of antibody that you produce. So you can see that it starts peaking at close to 2 weeks. So it takes some time for the response the first time you're exposed to an antigen. So it takes some time before your antibodies level reaches a certain value. Now you see here on the right side, if you're exposed again now to the same antigen A here, your response will be not only faster, so you reach the same level here. Instead of in 2 weeks, you reach it in 4 or 5 days. Okay. Not only that, but also this response will be also much stronger. You get 3 times as much antibodies produced, which means that if you're exposed, the presence of memory cells there allows you to fight that specific antigen and therefore that specific bacterium or virus that was containing that antigen in a much faster way the second time you're exposed to that. You can see how this does not affect the exposure to other antigens. So this could be the antigen for a different virus. The response in that case would not be affected by the previous exposure to another virus. Okay. Now this is all done. So all of these is done through what we call clonal expansion and selections. So what happened there, if you think? This is the situation you have before you get any activation of the antibody-mediated response. These are your B cells in your body. You have billions of them, okay? All with different receptor, ready to recognize different possible combination, therefore different possible antigen. But now you have, there are certain bacteria has entered, right? And through the stimulation, so first it was phagocyted by dendritic cells, macrophages, and do they present it to the CD4 T cells, which then do present the antigen to certain type of B cells. So now you have that only these B cells, not the other, start expanding, start producing more and more of these cells, right? Through clonal selection. So you have selected a certain clone that then will have expanded. And these are the cells. So now your immune system would have many, many more of these cells compared to the others, even though the starting at the beginning, this was at the same level. So, and these will lead to the formation of a very much enlarged population specific for that antigen and therefore for that bacteria or viruses, with some of these cells becoming plasma cells fighting that organism and some becoming memory cells, which then will be ready to fight specifically for the next. You'll see later on at the end when we talk about how the variation of this receptor is produced, how at this level 2 there is more variation being produced by hypermutation. Now, how do antibody act? They act in different ways. So just by binding to some molecule like toxin, they can neutralize them. Just by binding them, you prevent them from binding to their specific target, right? So just binding them, you remove them away. Other things, they can cause agglutination of bacteria by binding them. So they have two binding sites there. That causes bridging between bacteria, agglutination. Basically, the bacteria cannot move anymore. You already blocked them. Some of them, they do stimulate the activation of the complement system. Do you remember those 30 proteins that you have in your blood system? And others do stimulate also the phagocytosis by other cells, by antigen-presenting cells. By macrophages and dendritic cells that stimulate them as well. Now, what we describe here is what is called the active immunity. So the stimulation of the production of antibodies by cells that were already present in the body of the person exposed to the antigen. The same principle can be used also to produce passive immunity. If you isolate these antibodies, you can actually do that and inject them on a person. Oftentimes, this is used, for example, to inactivate the venom of some snakes, for example, right? You can take the antibodies, basically the serum from horses that have been exposed to the same venom, and use that. In that case, you would talk about passive type of immunity. Now, there are drugs that do affect these antibody-mediated pathway. And one example is cyclosporin A, for example, a drug that is used, for example, before a person undergo transplantation. So you want to suppress the immune system, and exactly the way they do it is by blocking the activation of the helper T cells. By doing that, you block the activation of the helper T cells, and therefore they won't be able to activate the B cells anymore against, in this case, the organ that's going to be transplanted, which the recipient of the transplant would take it as a foreign organism, right? So by doing that, you block the immune response against the new organ, and you hope that this organ can slowly get accepted by the recipient of the transplant. I'm going to stop here. I'm going to resume. This is the first part. Next part, we'll talk about the cell-mediated immunity